So I want to talk to you about the European Bank for IPS cells. So EBIS was started in an original funding phase running from 2014 to 2017. And we are currently in a second funding phase aiming for self-sustainability long term. And we also work with additional research projects to help them in their own sustainability goals. Next slide, please. Yeah, next, please. So as a brief introduction for all, what are induced pluripotent stem cells? As we are the European Bank of Induced Pluripotent Stem Cells. So IPS are derived from samples that have been donated by patients and volunteers. And here I also just want to really thank Helen because I think you really highlighted the role of patients and study participants in research. And without you, IPS research would just be completely non-viable. So essentially what happens is when an individual donates a tissue sample, normally a skin biopsy or a blood sample, they can then be, those cells can be reprogrammed to what we call pluripotent, which essentially means they can be differentiated into any cell type in the human body, allowing us to make disease-specific models and cell-specific models for research purposes. So for example, we could collect a tissue sample from an individual diagnosed with Alzheimer's disease, reprogram it and differentiate it to neurons and enable us to have a disease in a dish, so Alzheimer's neurons in a dish, which we can then use to look into how the disease has developed, how it progresses, and then of course moving into development of new treatments. Next slide, please. So what happened before EBIS was established? So patients or the general public would donate tissue samples either to a biobank or to a research team or a collaborative research project, and a research group would generate an IPS cell line. They would then be using that cell line, they might be able to publish some data outputs, and if the cell line is published, then it is findable, but maybe not so easy to be findable. The cell lines could be shared if that organization wants to share them, but it would require other researchers to really go through really laborious one-on-one negotiations about technical transfer of the cell line. Once EBIS was established, what that research group can actually do is deposit the cell line directly into EBIS. We can store it safely. We can expand it and bank it to make more of it. We can qualify to make sure that the cell line is of a very high quality. And then we can distribute those cell lines to researchers, both non-profit and for-profit researchers, for research use only. Meaning that any cell line that is used, any cell line that has been used in the original research study is also then widely available, meaning that other researchers can reproduce the original study that was done and also, of course, do new studies as well, meaning those original IPS cell lines are findable, they're accessible, they're interoperable, and they're reusable. And here, EBIS's really core goals is to increase reproducibility and replicability of biological research, particularly disease-associated research. Next slide, please. So where are we at the moment with EBIS? So to date, we've collected around 900 IPS lines, actually with more coming in at the moment. And those IPS lines have been generated across more than 30 different clinical studies. They were generated by more than 20 different organizations across Europe and North America, although we do accept cell lines from worldwide. And we collaborate very, very heavily with different research projects, including some other IMI projects such as Adapted and STEM Bank. Also HIPSKI was a very big project that we collaborated with, but also smaller research projects as well. So it's not just the big projects, even really small research projects can have a very, very big impact. Next slide, please. And what we really aim to do is to provide cell lines and provide essentially disease models across a number of different diseases, with neurodegeneration and neurological diseases being a very large focus for us. So you can see particularly on the left here, we have the breakdown of disease lines available through EBIS. So we have some neurological diseases, so migraine, pain, epilepsy, neurodegenerative diseases, Huntington's disease, ALS, FTD, and also Alzheimer's disease. And within each one of these disease groups, we really try and cover some different known genetic variants as well. aiming to be able to provide the disease models that researchers really need for their research purposes. We also try and provide a really good range of control lines so that researchers can have access to the control lines that they really need, including aged match lines, sex match lines. And if you look on the right-hand side, for the healthy control lines, so these are IPS lines that have been generated from individuals who, when they donated a sample, hadn't yet been diagnosed with any disease. We really try and cover a wide range of age ranges, and we have a nearly 50-50 split in terms of biological sex across male and female donors. Next slide, please. So it was one example here, and I mentioned earlier that we had collaborated with the project IMI Adapted. So IMI Adapted was a project aiming to study the pathology and progression of Alzheimer's disease, and they were specifically interested in this gene variant. So ApoE is the gene, and there's a number of different variants that are associated with the risk of being diagnosed with Alzheimer's disease and the rate of progression. And IMI Adapted engaged with EBIS very, very early on in the project, and we worked very closely with them to help them with their research goals. So essentially what we did was IMI Adapted asked us to generate gene-edited lines from 4 different donors. So 2 healthy donors, male and female, and 2 Alzheimer's disease patients, male and female. And from each of those donors, we generated original IPS lines, and then we gene-edited those lines so that we had a collection of lines from each, carrying the different ApoE variants. Those lines were then shared directly back to the adapted partners, who used them within their own research, differentiating, characterizing the cell line, the neurons that were coming out of it. And in parallel, we were able to make those cell lines available directly to other researchers. So on the behalf of Adapted, this had a really great impact for them. It meant that they didn't need to worry about setting up standard processes for things like cell line generation, cell line quality control, or distribution. They could just really fully focus on the research question at hand. And from our perspective, it meant that we were able to make these lines, which were deemed really, really valuable in terms of their scientific interest and the novelty, available to other researchers straight away. Next slide, please. So as a kind of brief overview of the scale of impact that this collection of these lines had on the wider research community, we have shared very high numbers of the lines. 3 figures worth of vials have been shared across all the different researchers. And you can see on the left-hand side that it's a fairly even split between distributing to academic and commercial researchers. We have also distributed them very broadly worldwide, both in Europe and North America, but also Hong Kong, Japan, Australia. And they continue to be of very high demand. We are aware of many publications that are coming out imminently for other researchers using these lines. And so from our perspective, these have really demonstrated a high impact, both in the non-profit and the for-profit research organizational sectors. Next slide, please. So looking at EBIS as a whole and the impact on research, so how do we help individual research projects? What do research projects that are using and generating IPS lines need? So they need a sustainable home for the IPS and the data sets that they're generating. What can happen otherwise is that at the end of a research project, the IPS cells remain sitting in a freezer in a research organization, and then they continue to be used internally. But really what that research project needs is a way to make the IPS lines very broadly, widely, and easily available to other researchers. They need support with sharing with other collaborators, and they also need backup in case their freezer fails or there's a technological failure. And they actually need to be able to focus on their own research outputs. So, really making the most of the research resources and time that they have. Next, please. So EBIS can really help them improve the sustainability of their research IPS lines and data. We can also then provide IPS lines at an industry level grade to other researchers. So really raising the quality of IPS research across the landscape. We can also help researchers with setting standards and best practice, and we work very, very closely with other similar groups in the IPS research field to really agree and set these standards for all researchers to be able to adopt. We increase accessibility to IPS research for non-expert users. So IPS can be slightly technologically difficult to use, and we really try and make it as easy as possible. We try and make the handling of the cell lines very easy, and we supply very accessible protocols all researchers can use. And through those methods, we try and accelerate research, also through service provision, so similar to what we did with Adapted, providing services such as reprogramming, gene editing, differentiation on behalf of other researchers. And that means then for researchers worldwide, any researcher has access to any of the lines within EBIS as long as they are within a research organization. The lines are clinically relevant and really high quality. And those researchers can then reduce the cost of their own research, because instead of generating brand new cell line cohorts, they're able to use cell lines which have already been generated, already been characterized, and maybe even already been published. They can repeat the data that has already been done, so increasing reproducibility. And we can also reduce wastage of resource due to the use of poor quality lines. And so here we really engage very highly and very broadly with researchers, both in Europe but also more broadly worldwide, to really understand their needs, their demands, and how EBIS can help and support them. Next slide, please. Now, we couldn't have done any of this if it wasn't for a public-private partnership, because we really aim to support both researchers from non-profit and for-profit organisations. The input of both public and private partners has been critical throughout EBIS, right from day one, in order to be able to set up processes and generate IPS lines, qualify IPS lines that really meet the needs of both groups. Obviously, both organisations in some ways have very different needs and very different focuses, and so that has really been super critical for us. As an example, looking at things like research groups depositing lines into EBIS. For the public partners, we observe that really we need to make that process as quick as possible, as easy as possible, and we should try and streamline it as much as possible. For the FPA partners, we realized that actually the really critical things around terms of use, the licensing, the intellectual property, and how that is tracked, how it is communicated. And so how we share that information about what intellectual property may be attached to the lines and how what a for-profit company needs to do in order to be able to use them. And so both of those things are very different, but very, very critical to the core processes. In our current funding phase, the public partners are helping us massively with developing new protocols, developing new cell types, and advising really common applications for academic users. And the FPA partners are really helpful in advising us what kind of differentiated cells are really of the highest use, what formats they are needed in, what level of maturation, what level of qualification. And this is really helping us in our EBIS 2 sustainability goals to derive new products and services for the bank. Next, please. And so here I just summarize the main outputs of EBIS, both things that we have already established and that we are now building upon, and also new things that we are working on. So in terms of the cells, we obviously have a collection of very high quality research grade IPS cells. We also within those have gene edited lines and a collection of different control lines. We use automation and upscaling approaches in order to be able to expand and bank and differentiate lines very efficiently. We are also working on the generation and distribution of mature derived cells and establishing further clinical networks so that we can help researchers who need IPS lines from a specific disease or genetic background in order to be able to access them. But it's not just the IPS cells, it's also the data that goes with them. So the richer the data is, the better the use of the IPS cells can be made. And so we collect, store and share the original data from the research team that generated the line in the first place. We generate quality control data, which is shared to the user. And we share some very high level non-identifiable donor data, and then any what we would term personal data sets, so things like genomic data sets or clinical data, we store in secure data archives, and they are available as managed access. And we also really try to support users however they need. We have, as I said, we have protocols widely openly available on our website. We have frequently asked questions that applies not just to EBIS, but generally to IPSC research. We can supply standardized transfer documentation, and we can also advise projects on some common ethical and legal challenges, which are very frequently observed in research projects. And we can really advise what those are and how to tackle them right from the beginning of the project onwards to prevent delays further on. And with that, I will finish up. So next slide, please. I would just want to say a huge thank you. Apologies. So here, EBIS2, we make the tools findable, accessible, and reusable. We improve sustainability for EBIS, but also for other research projects. We improve reproducibility and we accelerate research outputs. And so I want to say a huge thank you to IMI and also to all the EBIS2 project partners who have been working so hard the last couple of years to help us reach our goals. And also to all the depositors who work with us, all the research organisations that are really supporting open science by making the cell lines and the data available so that we can share them with other researchers. And the last slide. There's some contact information here. If anybody would like to get in touch, please do. And there's lots of information and support on our website. And you can also contact us directly by email or on various social media channels. Thank you very much.